Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Frank Furnari, Professor of Medicine, University of California, San Diego, and the co-director of SSCI. Today, we will explore Frank and his team's work in glioblastoma, tumor modeling, and therapeutic strategies. So welcome, Frank.
Thank you, Deb. It's really great to be here. I really appreciate the invitation and an opportunity to talk about our work in brain cancer.
This is great. I'm so interested in what you're doing. Let's first start with, what is SSCI?
So that is the Sanford Stem Cell Institute, under which there are numerous centers and one particular program. I'm the co-director of the Brain Tumor Program that is part of SSCI, or the Sanford Stem Cell Institute. We are an institute at UCSD that has a focus on stem cells from a variety of directions, whether it be understanding stem cells in aging. And in my case, I am interested in trying to understand cancer stem cells and how they play a role in brain cancer initiation and evolution.
So glioblastoma, that's one of the things that you study. And I guess it's known for really being very complex and resistant to any kind of treatment. It's a horrible cancer. What makes this cancer so challenging to treat?
Yeah. This cancer, maybe more than any other cancer, has heterogeneity, or really robust heterogeneity. And what that means is there are vast differences between individuals. So that would be intertumoral heterogeneity. And within an individual tumor, we have what's called intratumoral heterogeneity. And heterogeneity just means differences. So differences in how the tumor appears across its landscape. So if you took a section of a tumor and you looked under the microscope, you would see the tumor is very diverse across what you see under the microscope. And that diversity lends itself to the resistance that we have for these patients. So we have different mutations throughout the tumor and something called transitional cellular states. So there are cells within the tumor that are very plastic, or they have what is like a chameleon appearance to them. They can transition from one state to another. And those states have different susceptibility to drugs. So it's trying to target a moving target, if you will. And so that has been our problem. Not only that, these tumors are very invasive. So surgically, the surgeon goes in there, he debulks as much as he can, safe total resections of the tumor. But invariably, there are cells that migrate pretty far away from the tumor, and therefore the tumor cannot be cured surgically. So we have a big problem. We have this invasive potential of these cancers, as well as these heterogeneous states that we really want to understand. How can we target each one of these heterogeneous states? Or is there a way to, let's say, synchronize the tumor into one state that is therapeutically sensitive? And that's something we've been working on quite heavily.
The transition to the different stages, is that pretty rapid, or can you give me a sense of time? Like, is it kind of a known pathway, or is it really random?
We really don't know in terms of time. So you can, and this has been done, you can take cells out of the tumor and isolate them and re-inject them into an animal maybe a few cells, and then the resulting tumor recapitulates the heterogeneity that you had in the original tumor. So this ability to transition between these states is very plastic. And it probably has a lot to do with where the tumor cell finds itself in the brain. Is it near a blood vessel? Is it near a neuron? Is it in an area of the tumor that is either rich or poor in oxygen content? So there is this aspect of the tumor microenvironment that influences these transitional states of the tumor. And likely interactions with neurons, we are starting to understand that in the old days, we think of GBM as a cancer that's in the brain, but it is actually a cancer of the brain, which means that we need to take into consideration how these tumor cells interact with their normal environment. And that normal environment are our neurons, glial cells in the brain. And this interaction builds like an ecosystem that we are just beginning to understand how that works. So the connections between normal cells and tumor cells can actually help the tumor to grow more rapidly.
And that was going to be my question about, where do the tumors most likely form? Is there a part of the brain, or is it just wherever these cancer cells land as far as the process, and then they kind of take root, if you would?
They can grow in different regions of the brain. Where they originate is still an area of investigation. There are zones in the brain called subventricular zones, and we think that there are stem cells in those areas that could serve as the cell of origin, let's say, for a glioblastoma. But once those mutations occur in those cells, the tumor cells can migrate in a variety of areas of the brain. So there's no one region of the brain that is more susceptible than another for the growth of these tumors. And then if we think about pediatric cancers, they actually have certain regions of the brain that you would tend to find them. Like there's a type of cancer called the diffuse midline glioma that occurs in younger individuals, and those are along the midline of the brain typically.
And why do we know why that is? Is that just something that is part of the development of the brain that they end up there?
That's an excellent question. So we think that might be the case. There are particular mutations that occur in those types of cancers, these diffuse midline gliomas. They occur in a protein called a histone. And we think that those histone mutations are critical for brain development. So it's no wonder that they're occurring in these regions of the brain. I mean, you see these tumors in the regions of the brain where that histone is really important for the development of normal brain.
That's interesting. So your lab utilizes some advanced models to study this, this cancer. Can you tell us a little bit about what these models are? And you kind of talked about the stem cell aspect here. Can you expand?
Sure. A couple of years ago, we developed a model platform, which we call Cancer Avatar. And the way it works is we took normal human stem cells, or what we use is induced pluripotent stem cells. And these cells are very plastic. You can manipulate them to differentiate or become other types of cells. So you can take an iPS cell and coerce it to become a neuron or an astrocyte or an oligodendrocyte. These are all types of brain cells. And so we thought, like, well, what if we put the mutations in iPS cells that we typically see in different categories of GBM? We could put them in there using a technique called CRISPR-Cas9 gene editing. The very popular technique. It won the Nobel Prize a few years ago, which has been opening many doors in research. So we use CRISPR-Cas9 to make edits or gene mutations in iPS cells. And then we differentiated these cells into what we think is a cell of origin for GBM, which is a neuroprogenitor cell. It's one of the cells of origin. And what we found was when we put those cells into a mouse's brain and waited about 3 to 6 months. Those NPCs, we'll call them, became very good. They developed into very convincing GBMs. So when we looked at the brains of these animals by microscopy, they looked just like a GBM. When we analyzed them for their sequence, they had the same mutations as GBMs. They had the same expression patterns as GBMs. So we were able to essentially make these mimics of human tumors, human GBMs, using human iPS cells. And we've done this for many different types or subtypes of GBM. And we've also done it for pediatric cancer. So very different brain cancer types, such as diffuse midline gliomas, another one called ATRT, atypical teratoid rhabdoid tumor. So this has really served us well to make a whole catalog of these different cancer types. And it's great because we're starting from the same human iPS cell. So everything, the genetic background of all of these brain cancer models is the same, except for the mutations that we put in. And so this has helped us eliminate some of the noise that one can get if you want to analyze patient samples. Every patient sample is going to be very different. They have their own genetic background. So it becomes a little difficult to try to find vulnerabilities or therapeutic vulnerabilities if you're looking at a dozen patient samples. But using our avatar system, because we have basically minimized the mutations that are in there, we have I think a cleaner system to do the initial analysis and find vulnerabilities, which we've started doing, and it's been working pretty well.
So the stem cell comes from a scratch of the skin or some sort of sample. How did you choose that donor? If I'm naming this person correctly, how did you find this person? Because they could get mutations. I mean, what's the science behind the choice?
So we started out with both a male and female iPS line that was commercially available. So a company used skin cells and reprogrammed them using available techniques to make stem cells. And they sequenced those individuals' iPS cells, and they were completely normal. And so we bought them off the shelf, and we used that as our starting place for making CRISPR-Cas9 mutations. So start with what appears to be a very normal individual. Either we have a male and female, as I said, and we can make the mutations using CRISPR.
So a couple of weeks ago, I think it was NIH came out and asked, talked about this transition from mouse models to, ISPCs or organoids or whatever the definition of that. Where's kind of that thought? And if you could kind of share because it seems to me that it's probably getting us closer to real life versus using rat or mouse. But maybe there's still a reason for the rat or mouse.
I think what we'll see is we won't eliminate the rat or mouse for experiments because they do give us information about survival. So if you want to, engraft a brain tumor into an animal and look at how long they survive on a therapy, that's vital information. But there's definitely an in-between model system we could adapt. So if you go to the other extreme, is growing cells in a plate and trying to treat those with drugs. And, you'll get some information, but it's definitely not going to be truly indicative of what would happen in a person. So I think the field is starting to come together and realize, we can make little brain organoids in a dish using iPS cells. And then to those, we can engraft patient tissue, patient tumors, our avatar models. So essentially, you can model a brain with tumors in them, little, these microscopic little brains in the dish, and we can analyze how well the tumors that engraft, how well they invade, do they respond to therapy, So we can mimic some of the aspects of a mouse or a rat model in a dish much better than we used to in the past. So I think taking all of this together, it likely will minimize rats and mice, but not completely.
So back to this kind of this transition to the stages of the tumor, how it kind of moves and adapt. By using these advanced tools or models, the organoids, will we be able to better understand maybe that pace or the different kind of transition phases?
I think so. So one of the things that we do is we can do single cell sequencing on either patient samples or our avatar models, and we can directly assess the transition states, the abundance of, there's typically 4 states that occur in these tumors. So we can assess that, we can quantify that, and we can apply different pressures to the tumor. If it's in vivo in the mouse brain, we can treat it with a therapy, and we can harvest the tumors over time. And then we can ask the question, okay, what do the transition states look like on therapy? Have we managed to corral the tumor into one therapeutic vulnerability state, maybe eliminate the other 3 states of the tumor? And can we do the same thing in a dish? So in these organoid models, can we treat with a drug and cause the tumors to become more sensitive to radiation? And does it track with a particular transition state? So I think we have a good handle on how to assess transition states over time and also on therapy.
How hard is it to diagnose brain cancer, glioblastoma? Is that something that, it is it really kind of advanced by the time people know when they go into the doctor? I mean, and do these advanced tools and models, will it help with diagnostic capability?
Yeah, so unfortunately, most individuals who get diagnosed with a GBM, it has caused something like a neurological symptom. They have a headache, or they have numbness on one side of their body. So these are symptoms that correlate with the presence of a GBM. Many other things, but that would be one thing that likely will be looked at. So typically you don't see a GBM forming in a person at an early stage. I wish there was a way to do that. I mean, there's been some investigators who think there could be a blood test for GBM, because if there's a mutation in the tumor, maybe that DNA will be shed into the bloodstream, and you can do an annual physical, and you can test for the presence of that mutation. It could be a panel of mutations, and they can test. We're not quite there yet, but there are definitely labs that are working on that, building up these panels of very typical mutations that one sees not only in brain cancer, but bladder cancer, pancreatic cancer. I mean, there are very typical mutations that with some refinement, we should be able to spot in peripheral blood, because DNA gets shed from cells when they die, and we should be able to detect that. So I think that's where the field is right now. There's really no easy way to know a person has a brain cancer until they come in with some sort of neurological abnormality.
You said today it's kind of the state of the art, if you would. You have a surgeon cut the tumor out, and then you still have to go through some kind of radiation or possibly chemo. I think you said that glioblastoma is very resistant to the radiation. Can you expand on that? How is your research trying to get around that or to try to address that?
So that's an excellent question. So we do know that GBM has resistance to radiation. So standard of care therapy consists of surgical debulking followed by radiation and a chemotherapeutic drug, typically a drug called temozolomide, which induces DNA damage, and hopefully the tumor cells will succumb to that. But we do know that tumors have different areas, once again, that are vulnerable or not to radiation. This gets back to these transition states. One particular state, which we know is not very sensitive to radiation, is called a mesenchymal transition state. And so what we've focused on and we have a paper right now being reviewed, is can we block the mesenchymal transition state? And we have a small molecule that is targeting a protein called BRD2, bromodomain containing protein number 2. And this small molecule diminishes the mesenchymal state in the tumor. And because of that, the tumor is more sensitive to radiation. So we're hoping to expand upon these studies. My big hope is that maybe we could push this forward to a clinical trial. So treating with a BRD2 inhibitor followed by radiation is what we've done in our paper. And we see an increase in survival of mice with our GBM models.
Is there stages that are not resistant to radiation that you can excite to that stage? I don't know if I'm asking that question correctly, but it's putting them in, putting those cells in the state where it becomes resistant or more resistant to radiation or chemo.
So the mesenchymal state of GBM10, that state definitely correlates with resistance. If we could dampen the gene expression patterns in a mesenchymal cell, they become more of what we call a proneural state. And that state is more sensitive to radiation. So that, I think, answers your question. We're finding ways to essentially, I'm not sure the word synchronize is the best word, but it seems to fit here, where we can move the tumor towards a more therapeutically sensitive state. So an additional piece of information is when patients are treated with standard of care therapy, they will respond initially. And then when they relapse, which is very typical, the tumor that comes back is much more mesenchymal in appearance, in gene expression. And it makes a lot of sense, right? So we know the mesenchymal is less sensitive to standard of care therapy. And so we're actually selecting for or driving the tumor towards that state. And that's what we see at recurrence. So we hope with the small molecule that we have that we can prevent that. We can prevent that transition to a mesenchymal state and sensitize the cells to radiation or keep them sensitive to radiation and chemotherapy.
Are there other emerging drug therapies that will also help? Are there things that are in trials right now that you might be able to use too in your experiments?
Yeah, I think the most exciting area right now in GBM or just brain cancer research is using CAR Ts. So these are chimeric antigen receptor expressed on T cells. And this technology has worked for other cancer types. So using a CAR T with a receptor that is engineered to express a mutation on like melanoma has worked really, really well. But the same approach for GBM has not. And we're starting to understand why. The GBMs tend to not have expression of antigens or novel antigens on their cell surface. So we need to somehow educate the CAR Ts to recognize perhaps some small amount of these novel antigens in the tumor. CAR Ts have a difficult time of getting into the tumor. So we need to understand why, what are the barriers to of the microenvironment of the tumor that prevents these CAR Ts from getting in. I think there's really good hope that a combination of therapies, like I mentioned with a BRD2 inhibitor, plus a CAR T inhibitor, and perhaps even a vaccine. So if we could sequence every patient's tumor and understand, okay, if there are mutations in the tumor, has that mutation created a novel sequence or peptide sequence or protein sequence that we can now leverage to make a peptide vaccine. And people are doing that. So I think all of these new approaches leaves me very hopeful. I mean, I've been working on brain cancer for many years, and I would love to see that we push survival out, many months, maybe years beyond where we are now, which is about 14 months on average. So yeah, it's been exciting to see the development of these new tools, these new approaches to try to treat brain cancer.
These vaccines that you're talking about, these kind of future vaccines, that would stop the reoccurrence, or is this something you get vaccinated, younger in life, or where would that be?
No, it would definitely be based on the tumor that a person has. So it would be personalized, what we call personalized therapy. These mutations would not exist anywhere else in the body, so there's really no reason to vaccinate individuals, at childhood or whatever. So we would have to sequence individual tumors and understand what mutations, the mutations that occurred in the tumor, what new antigens, if you will, have arisen, and we can leverage that to make these vaccines or make these peptide vaccines. So my vaccine, if I had a brain tumor, would be different than the vaccine for you. So it's definitely on the horizon. There are people working on this, and I think within maybe five to 10 years we'll be seeing a bit of this being implemented in the clinic.
So I think you said, the glioblastoma, the cancer in the brain, not of the brain. And, so much that we're finding now as others study different diseases, whether that be ALS or dementia or that, is that there's a relationship with other parts of the body, the blood-brain barrier, cardiovascular, maybe immune system, gut biome. These cancers, could they be kind of manifesting in other parts of the body and then just having preference in the brain? I don't know. Where's that science? Where's that research?
So GBM and everything we've discussed today are cancers that start in the brain. So their origin is in the brain. There are other cancer types that migrate to the brain, such as metastatic breast cancer, lung cancer. These are cancers that, we can treat the primary source of the cancer, the lung or the breast. And unfortunately, the recurrence of those diseases tends to happen in the brain. It's a site that those cancer cells can migrate to and thrive quite well. So those cancers are, they're obviously different than a glioblastoma, and their susceptibility to drugs is different than glioblastoma. Now, glioblastoma is, it's so successful as a tumor in the brain, and I think that's because it really interacts with its normal counterparts, cells around it, and as we mentioned, interactions with neurons. We really didn't appreciate this up till a couple of years ago. There's a whole new field that's emerged called cancer neuroscience, and people are finally understanding. The neuroscience. If you went to a neuroscience meeting that focuses on ALS and Parkinson's and all other types of dementia, you probably would not see a lot of brain cancer research at such a meeting. But now I think the field is changing. We understand we need to talk to each other because we need to understand neural stimulation, neuron interactions, and how those get co-opted by brain tumors. And it's been pretty well established now that neurological impulses can actually stimulate the growth of a brain tumor. So understanding what those signals are from a neuron to a brain tumor is a very, let's say, hot field right now in brain cancer.
Yeah, that's really interesting.
Yeah, and we can only understand this by having conversations with our colleagues in the neuroscience field.
Yeah, that's really interesting. Why study, brain tumors? What was it that sparked your interest early in your career, and what was that journey?
So I started studying glioblastoma in 1993 when I came to San Diego from the University of North Carolina, where I was a grad student at UNC, and I was studying something completely different. I was studying DNA tumor viruses, one of which was Epstein-Barr virus. And I decided as a postdoc, I really wanted to understand how cancer forms. It was a field that was really growing at the time because we were starting to understand the role of genes called tumor suppressor genes. So all cancers have fundamentally two types of genes, oncogenes and tumor suppressor genes. Oncogenes are like the gas of the car, and tumor suppressor genes are like the brakes. And you typically have mutations in both types to get a cancer to form. And at the time, when I was looking for a postdoc position, there was an investigator here in San Diego. His name is Webster Cavaney, and he is credited with first describing tumor suppressor genes. So he was looking at a pediatric cancer called rhabdomyosarcoma, RB for short. And I'm sorry, he was looking at retinoblastoma, which is a tumor of the eye. And he found that there were chromosomes that were typically lost, or parts of chromosomes that were typically lost in these kids in their tumors. And he did further analysis, and he was able to define regions which he hypothesized had genes which controlled the growth of these tumors. But they controlled the growth only, or they prompted the growth of these tumors only when they were mutated or lost. So those portions of the chromosomes were missing. And one thing led to another. They were able to identify that the gene was called the retinoblastoma gene. And this led to a whole new field of science of finding tumor suppressor genes in all types of cancers. And at that time, for glioblastoma, we knew that there were regions on chromosomes which were consistently lost in these tumors. So it's like a 30,000-foot look at the tumors. We could say on chromosome 9, there is a missing region in most GBMs. On chromosome 7, there's a region missing in most of the cancers, but we didn't know the name of the gene. We didn't have it isolated, and that was an exciting thing for me because I really wanted to get in and try to understand what these genes were. And so during that time, in my early part of my career, I had a hand in characterizing one particular gene, which is mutated or lost in 40% of glioblastoma. And that's called the PTEN tumor suppressor gene. It was identified by individuals at MD Anderson, and I then classified its role as a tumor suppressor gene. I showed that if you put it back into tumor cells, that it suppressed the growth of those cells. So that was the first illustration that this was a bona fide tumor suppressor gene, that it was vital to regulate the growth of glioblastoma. And so that was the launch of my career, if you will. From there, I kind of like, oh, this is great. We're starting to understand how these cancers arise, what are the mutations in particular genes. Then the human genome was sequenced, and we then had a really firm understanding of mutated genes in many cancer types, notably GBM, which was one of the first cancers to be completely profiled for its mutations. And so, I feel humbled to be at the very beginning of this field of completely molecularly understanding glioblastoma. And it's an exciting time to be in this field because I think we're finally starting to leverage all the things that we've learned to turn them into therapy for patients.
Yeah, that's fantastic, and I bet you, as a young researcher, that was pretty exciting to be a part of that, and I can see where that was the kind of the pivot point. So you talked about all the information that we're kind of gathering, and and it seems to be kind of accelerating up. Bioinformatics, how maybe even AI, how is this kind of changing what you're doing to accelerate that knowledge and then get to therapeutics?
Sure. Bioinformatics is indispensable now in most labs. I mean, when I started my career, I was mostly like a bench scientist. We call it a wet lab scientist, where we do experiments in plates, in animals. But now, when we have the ability to sequence tumors at very high resolution, we can understand the mutations at the single cell level. Well, that requires a really good understanding of bioinformatics, because you generate these large data sets that you look at like, oh, what does this all mean? So I have been very fortunate to have some great trainees in my lab who are much better at bioinformatics than I am, but they can make sense of these large data sets and understand, try to deconvolute the information and understand these transition states. What are the mutations in the tumor that we could informatically understand and then define new therapies towards. So bioinformatics is key, I think, for any cancer lab right now. And then for AI has been another revolution, in our research. You can use AI. There's one particular program called AlphaFold, and this came about only a couple of years ago, and it was just transformative in how we approached protein science. So what it does is it can take the amino acid sequence of any protein, and it can predict how that protein folds. And that's an unbelievable leap in our ability to understand protein function. What we used to do is you had a protein, then you wanted to see how it folded. Well, you've got to express that protein in, let's say, bacteria or yeast, isolate it, and then do some sort of X-ray crystallography to understand how it folds. And that is a very long process, doesn't always work. But now AlphaFold can do the same thing on your computer in a fraction of the time. And in most cases, it's absolutely correct. I mean, people have to validate what they learn from that. But once they know the structure of a protein, they can say, aha, I know now the protein has this little pocket in it. I can probably design a small molecule that will go into that pocket and block its activity. So that, as I said, is just revolutionary for the field. And these tools, these informatic tools, are really changing how we approach the whole problem of cancer and treating patients.
Yeah, I'm sure your lab today is very different than what you worked in 1993. What are the challenges in having a lab today? Is it time? Is it people? Is it money? Is it what what do you see are kind of the challenges as you're trying to accelerate this knowledge to an understanding to get to real solutions?
Well, I think money is always a challenge for any investigator right now, any time in history. So, yeah, that's a constant. But I think one challenge we have, as a PI, as someone who's running a lab, is really spotting talented people. And this probably goes for any business, right? So you want to have the most motivated individuals in your lab, people who want to come in there and just really hit the ground running and really dedicate their energy to doing the science. And I think I've been good at that, not 100%. So I think over time I've learned to identify those individuals. You can never teach motivation. The person either has it or they don't. And then just spotting that early on is, I think, a big key to running a successful lab.
You mentioned money. I'm kind of curious because back in 1993, things were more done with more people and tissue samples and such. Are labs getting more expensive because of the technology, or is the trade-off kind of balance? Where, give me some idea of, because sometimes people hear, oh, well, we have all this technology and you don't need the people, and so it should be cheaper and should be quicker.
Yeah, science has definitely gotten more expensive. Anything that has to do with sequencing, especially if it's single cell sequencing, those types of experiments cost tens of thousands of dollars just to take a tumor and sequence it at the single cell level and then, figure out the constellation of mutations. Very expensive. The instrumentation that we use to do this is also very expensive. It's not unheard of for a sequencer to be $300,000 or an instrument like a mass spec instrument to look at the complement of proteins in a cell could equally be $300,000-$400,000. So these are really big ticket items, and then keeping them in working condition. So what I have always been very surprised by are these equipment costs. So every year you want to keep your equipment running well, so you have to get the companies will sell you these annual subscriptions to come out there and take care of the equipment. That itself could be $20,000 to $30,000 just for a single year for one piece of equipment. And if you have 10 pieces of equipment in the lab, we're talking a very big ticket item just to keep things running and running smoothly. So yeah, I think science has definitely got more expensive. I mean, mouse costs, if you're doing those sorts of experiments, could be very costly as well.
Yeah, I think that's a good discussion to have with the public or possible investors that don't typically invest in research, because I think there's there's investors that want kind of on the product side. Are we quicker to, we're already over the chasm, let's get things to market. The research will happen, but as technology advances, I think it's important to have that discussion.
Absolutely. Because a lot of what I mentioned, the government won't fund. It's really hard to buy equipment through government funds. So private investment is really essential right now. So having someone who really wants to invest in your science, but understand, hey, he or she needs this new sequencer that will, blow open the doors on results. That is certainly key.
Yeah. Yeah, there seems to be a business model there for somebody that would want to do that, depending on whatever came out of that and whatever research or therapeutics came out of that. That could be a lot of leverage for them. So how has, you talked about, you've kind of grown into being a much better identifier of talent and such. What other things have, kind of your leadership, how has it, how has it changed over the years? And what, you're kind of in this, kind of crazy time. It's a, what are you learning as far as that leadership in today, and what are you telling people in your lab?
Yeah, I definitely don't want to be a downer to people in the lab because of what's our current environment. Because science to me is just so exciting, and to be able to think and ask these really important questions that no one's asked before. I mean, that's the true essence of science. It's this discovery and putting your mark on something to move the needle forward to help people. So I do my best to help them see that we are doing a noble thing. Doing research and trying to understand disease and try to come up with better ways to tackle, whether it be brain cancer or pancreatic cancer or any type of cancer or neurological disease, is in the best interest of the general public. There'll never be a way to overlook that. That is, I think, the absolute truth of what we do. So I try to be a cheerleader at times, but I'm also a realist as well. I'm pragmatic. I think I'm pragmatic. And I tell people, when you're thinking how to do your experiments, think wisely, think of the appropriate controls and everything that's needed to give the best readout of the question you're trying to address. And it's called, working smart, because funds are not as plentiful as they used to be. So we have to be good stewards of the money that we have. And I think that's gone over pretty well. My colleagues all around me take the same approach, and people are receptive to that.
So I spent this week at the Milken Global Conference. It's an annual conference. A lot of people, investors, leaders in policy and science. And what I walked away with is there is optimism. First of all, there's a belief in science. There's a belief in the need for research and a very strong need. And even if people don't quite understand all the ins and outs of it, I think there is high hope for new technologies such as AI or bioinformatics to help get things to patients quicker. But one of the things that was really kind of encouraging to me was the discussion around different kind of collaborations and how do we get this funded and I think there was a lot of talk about what could these be and how do we fill some of the gaps. How are you kind of, and maybe you've already done that, and maybe your portfolio has some diverse funding sources there. But, is that one of the pivots that you're kind of looking at is to, what kind of new partnerships can we put together to do this?
Sure. Yeah. Now you hit the nail on the head. So I will admit I'm not a tumor immunologist. I'm more of a cancer biologist. So if I want to understand how, GBM evolves, I need to know more about the immunological state of the tumor. And so I have a great partnership with an investigator at the Salk Institute. Her name is Sue Case. She's a fantastic tumor immunologist. So we're putting together a grant right now. That's just kind of one example of how partnerships really advance the science. I think they're synergistic in many instances. Other examples, one of my colleagues here is a bioengineer. So he knows how to work with different materials and understand forces on tumor cells and how that affects their migration. Once again, not an area of expertise of mine, but I understand how tumor cells form and different mutations and different subtypes of GBM. So together we've had a wonderful long run of working together. Several grants have been funded through our partnership papers, etc. So it's those sorts of examples of reaching out to individuals that really complement your skill set. And then a third example is, since becoming the co-director of the Brain Tumor Program here at the Sanford Stem Cell Institute, I've been putting on a monthly brain tumor meeting. It's actually called the Brain Tumor and Neuro Restoration Meeting. It's held once a month in my building, and I have invited people from all walks of life. They could be neurosurgeons, neuro-oncologists, engineers, natural compound, people who work at Scripps Institute of Oceanography, you name it. And I invite them all to take turns presenting their science. And it's already led to several collaborations amongst this very diverse group of people. And the reason I did that is I really felt that if you've got everyone in the same room is a cancer biologist, then you're all singing the same song. You need this diversity of thought in the room to approach a question. You need an engineer to come up with a new way to deliver a therapy to a tumor. Someone who knows how to work with lipid nanoparticles for drug delivery, or you need someone who is perfecting a surgical technique that maybe you can use on animals to ask a specific question. So that has, to my great satisfaction, we've only been at it about a year, having this monthly meeting. It's worked out really well.
Yeah, that's wonderful. Maybe in the future you can invite some health minded investors to take part too, and maybe they're interested in buying some of that equipment, or
Yeah, that's a great idea. Have them come.
Yeah,
we have pizza.
Yeah.
So they'll come if there's pizza, I hope.
Exactly. Yeah, exactly. So looking out 10, 20 years, you pick the horizon. What are some of the most exciting breakthroughs or, significant knowledge gaps that will fill that you're really excited about?
Yeah, without a doubt, this cancer neuroscience is the big field right now, the big emerging field that we're all trying to understand. Why does a tumor cell next to a neuron have a particular transitional state? Getting back to our original discussion, these transitional states are really important to understand. Why does a tumor cell next to a neuron have a specific transitional state? We need to understand that. And what does it mean? Can we disrupt that? And we have data now that we've been able to, one of my students has generated that when he looks at a tumor section and he looks at cells nearest the edge of the tumor near neurons, they're very different than the tumor cells in the core of the tumor. So understanding these neuron cancer interactions is key. And then are we going to be able to repurpose drugs that could actually work on the neurons or block interactions with tumor cells? That is maybe the next step here. I know there's people working on that. And I think, once again, understanding the immune system and how can we make it better adept at tackling the brain tumor? Why do brain tumors become so privileged? So they don't have T cells infiltrating that will tackle the tumor. Understanding that is also very key. And I think we're getting pretty close to making some large headway there. If we understand it for a GBM that's mostly mesenchymal, then the next step is to try to understand it for a GBM that's more maybe proneural in its gene expression. So there's a bit of work yet to do, but I think we have the tools in hand that within 10 years, maybe 20 years, there'll be significant changes in how brain cancer patients, in particular GBM patients, are managed.
That'd be wonderful. So, Frank, as we wrap up here, how do people get in contact with you? Maybe, investors that want to come to your collaborations and fund you, or just people that want to work with you. How do they get in contact?
I'm so easy to find on the internet. Just type Furnari Lab. You'll bring up my website there, and there's also an X link in there as well that one can find and follow us. And I also have a LinkedIn connection as well. So happy to connect with anyone who's interested in our science, and they want to email me at ffunari@ucsd.edu. That's also totally fine.
That's wonderful. Well, it's been a real pleasure, and I really appreciate the time you've taken out of your busy day. You've got important work that you do. So thank you so much.
Thank you so much, Deb. And one last thing, if I might say, this is Brain Tumor Awareness Month. So bring it to the public. Everyone needs to know how much emphasis we need to put on studying brain cancer, both in adults and pediatrics.
I appreciate that. We'll get this out very quickly.
Deb, thank you so much.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.